It's good to have you all back to Food Catalyst. Don't forget to join Telegram and the others, then you will miss an opportunity to participate in quizzes and any other updates. Let's dive back into the video. First question. Most abundant carbohydrate in the biosphere is dash. The options are starch, glycogen, cellulose and glucose. Hope you all know that except glucose, the other options given are polysaccharides. What are polysaccharides? Polysaccharides are complex carbohydrates made up of long chain of monosaccharide units. At least 10 monosaccharide units should be there to term it as a polysaccharide. Polysaccharides can be classified into several types. Like based on the composition, polysaccharides can be classified into homopolysaccharides and heteropolysaccharides. Similarly, based on their functions, that is the role they do, polysaccharides can be classified into several types. I have given two of the fundamental classification of polysaccharides based on their roles they do. One is structural polysaccharides, the other one is storage polysaccharides. Structural polysaccharides are carbohydrates that provide support, strength and structure to cells, tissues and organisms. The examples are cellulose and chitin. See, cellulose is the prominent structural polysaccharides found in the cell walls of plants. That means in all the vegetations, the structural polysaccharides is cellulose. And also chitin. Chitin is a structural polysaccharides found in the exoskeletons of arthropods such as insects, crustaceans, and even in the cell wall of fungi. Chitin is the structural polysaccharide. When it comes to storage polysaccharides, these carbohydrates that serves as the forms of energy storage, allowing organisms to store excess glucose for later use. The examples are starch and glycogen. Starch is the major storage polysaccharides in plants. Glycogen is the storage polysaccharides in animals, including humans. Now you all know all the options, okay? Could you able to find the answer? The right answer is cellulose. Cellulose is found in large quantities in plant biomass. As I already told, all the vegetations in major composition is cellulose. That is including trees, grasses, okay? It is estimated that cellulose make up to 33% of all plant matter, making it as the most abundant organic compound on earth. Even though cellulose is abundant in nature, it's worth noting that humans and many animals cannot digest cellulose as they lack the enzyme called cellulases, which is necessary to break down the complex structure cellulose. While animals themselves do not produce cellulose, but some animals have symbiotic relationship with microorganisms that produces cellulase on their behalf. These microorganisms such as bacteria and protozoa reside in the specialized compartments within the digestive system of these animals and help break down cellulose into digestible compound. The digestive systems of ruminant animals like cow, sheep and goats. These animals have specialized stomach called rumen, which contains a diverse community of cellulolytic bacteria and protozoa. These microorganisms produces cellulases that enable the breakdown of cellulose from the plant material consumed by the ruminants. So the right answer is cellulose. Come on, let's move on to the next question. The carrier of endogenous triglycerides from the liver to the peripheral tissues. Look, to answer every question, definitely you should have some basic knowledge about each and every words in the question. In this question, you should know about the meaning of what is triglyceride. In that, you should have a knowledge about what is endogenous triglycerides. So something is going to carry these endogenous triglycerides from liver to the peripheral tissues. The options are VLDL, LDL, HDL and chylomicron. All the options given are lipoprotein. To answer these questions, you should know what is the function of liver with respect to lipids and what is the term endogenous triglycerides and what is peripheral tissues. Peripheral tissues means the parts other than the central organs of the body. It includes muscle tissues, nerve tissues, etc. On looking into the liver is involved in the metabolism of all three macronutrients like glucose metabolism, lipid metabolism and protein metabolism. This basic information is enough with respect to liver. Now, what are lipids? Lipids are a diverse group of organic compounds that is insoluble in water but soluble in organic solvents such as alcohol, ether and chloroform. Here are the main types of lipids: fatty acids, triglycerides also known as fats, phospholipids, steroids, waxes and glycolipids. Cholesterol comes under the category of steroids. The chemical composition of any dietary fats including oil will be these lipids one. If you take groundnut oil, its composition is 100% fat, that is 100% triglycerides. Now we see how these lipids are metabolized. Lipid metabolism involves the process of synthesizing, storing and utilizing lipids within the body. It can be broadly categorized into two main types. One is exogenous lipid metabolism, the other is endogenous lipid metabolism. Exogenous lipid metabolism begins with the consumption of lipids in the diet. These lipids can include triglycerides, nothing but fat, cholesterol and various fatty acids present in the foods such as oils, meats, dairy products and nuts. In terms of endogenous lipid metabolisms, it refers to the synthesis, storage and utilization of lipids within the body, particularly in tissues such as liver and adipose tissues. Lipids synthesized in the liver through endogenous metabolisms primarily originate from various precursors such as excess glucose, proteins and fats. Now you all can understand that being triglycerides as a lipid, it can be undergo two pathways. One is endogenous triglycerides and other is exogenous triglycerides, which are all mentioned in the question. So through endogenous metabolism, once a lipids, either triglycerides or cholesterol is synthesized in the liver, they will pass through series of steps which involves lipid assembly, protein incorporation and maturation process. After this process, a new particle is created which is nothing but lipoproteins. So lipoproteins are complex particles that consist of various types of lipids and proteins. If varied lipids and varied proteins means, then various types of lipoproteins will be there. The major type of lipoproteins in our body includes chylomicrons, VLDL that is very low density lipoprotein, IDL that is intermediate density lipoprotein, LDL that is low density lipoprotein and the remaining is high density lipoproteins. These lipoproteins plays an important role in maintaining lipid homeostasis and cardiovascular health. Synthesis and secretion of VLDL particles. As we all know that the liver is responsible for the synthesis and secretion of VLDL particles. VLDL contains triglycerides, cholesterol esters, phospholipids and apolipoproteins. These VLDL particles transport endogenously synthesized triglycerides from the liver to peripheral tissues where they can be used for energy or storage. This lipolysis removes triglycerides from VLDL, leading to a transformation of VLDL into IDL, nothing but intermediate density lipoprotein. Further, lipolysis of IDL result in the formation of LDL particles. These LDL particles are smaller and denser than VLDL and IDL. So, LDL primarily transport cholesterol from the liver to peripheral tissues throughout the body. So, as a concluding point, VLDL particles transport endogenously synthesized triglycerides from liver to peripheral tissues. LDL primarily transport cholesterol from liver to peripheral tissues. So the answer for the question is VLDL. The next question. Specific dynamic action of protein, fat and carbohydrates is in order. To answer this question, we have to know what is specific dynamic action. Shortly, we can call it as SDA. The specific dynamic action is also known as thermic effect of food, or also otherwise called as dietary induced thermogenesis. These terms refers to the energy expenditure required for the digestion, absorption, transport and storage of nutrients following food consumption. Each macronutrients like protein, fats and carbohydrates has different SDA values, which reflects the energy cost associated with metabolizing the nutrient. Let's see protein. Protein has the highest SDA among the three macronutrients. It requires the most energy to digest, absorb and utilize. Approximately 20 to 35% of the energy from the protein is utilized during its metabolism, making it is the most thermogenic macronutrients. This high SDA of protein is due to the complex structure of amino acids and the energy intensive processes involved in the protein synthesis and nitrogen balance. Now carbohydrates. Carbohydrates have a moderate SDA compared to protein and fat. Around 5 to 10% of the energy from the carbohydrates is utilized during digestion, absorption and storage. The SDA of carbohydrates depends on various factors such as the types of carbohydrates, that is simple or complex carbohydrates. Even its fiber content matters. The fiber-rich carbohydrates may have a slightly higher SDA due to its additional energy required for fiber breakdown and fermentation in the gut. Lastly, fat. Fat has the lowest SDA of the three micronutrients. Only about 0 to 3% of the energy from the fat is utilized during its metabolism. This is because fats are highly energy dense and requires minimal energy for digestion and absorption when compared to proteins and carbohydrates. However, it's important to note that not all fats are metabolized equally, and certain types of fats such as MCTs, that is medium chain triglycerides, may have a slightly higher SDA due to their unique metabolic pathway. So in the summary, the right answer is the order of specific dynamic action is from highest to lowest. Protein is greater than carbohydrate is greater than fat. Next question. Coconut oil contains dash chain triacylglycerides. Look, here is a mistake in the question. It should not be triacylglyceride, it should be triglycerides. The options given are short, long, medium and very long. Could you able to guess the answer? Let's see the explanation. Here is a typical fatty acid composition of coconut oil. Coconut oil is composed predominantly of saturated fats, accounting for about approximately 80 to 90% of the total fat content. Coconut oil also contains unsaturated fats, vitamins and other trace elements. Among the saturated fats, lauric acid constitutes around 45 to 50% of the total fat. So it is a major constituent. Lauric acid is a medium chain fatty acid. So on seeing the other fatty acids, lauric acids and caprolic acid together gives the nature of the coconut oil as medium chain fatty acids. So the answer is medium chain fatty acid. That is medium chain triglycerides. Next question. Human cannot synthesize vitamin C because of the absence of the enzyme. The enzymes given in the options are L-gluconolactone oxidase, glucokinase, glucose oxidase, L-gluconolactone reductase. Can you find the answer? Let's see the explanation. First, what is vitamin? Vitamins are organic compounds found in very small amounts in food and are required for normal functioning. In fact, it is very much important for survival. It is a very common knowledge that unlike the macronutrients, vitamins do not serve as an energy source. Rather, they assist in energy yielding reactions and facilitate metabolic and physiologic processes throughout the body. And one more important thing, that vitamins do not undergo the same process of digestion as macronutrients. Instead, their metabolism involves a series of complex processes such as synthesis, absorption, activation, and enzymatic reactions. Further, both the gut microbiome and enzymes in the gastrointestinal tracts plays crucial roles in ensuring that vitamins are available for body's essential functions. So, in order to utilize the vitamins properly, we should maintain a good gut microbiome and keep the gastrointestinal tracts healthy. However, there are certain vitamins that may have challenges related to absorption or utilizations in the body due to the various factors. One such vitamin is vitamin C. Because humans, along with certain other primates, for example guinea pigs and some bat species, lacks a functional form of the enzyme L-gulonolactone oxidase, that is GLO. This enzyme is essential for the biosynthesis of vitamin C. It's widely recognized that the genetic mutations that led to the loss of functional GLO in humans occurred millions of years ago, likely during the evolutionary process. As a result, humans are unable to produce vitamin C internally and must obtain it from dietary sources such as fruits, vegetables, and supplements. So once we take vitamin C through dietary means, how it is absorbed? The absorption of vitamin C is nothing called ascorbic acid. The absorption of vitamin C involves active transport mechanism in the small intestine. The specific transporters such as sodium dependent vitamin C transporters, shortly called as SVCTs. This transporter facilitates the uptake of vitamin C into enterocytes, that is intestinal cells. This active transport ensures the efficient absorption of vitamin C, allowing it to enter the bloodstream for utilization in the body's essential process such as antioxidant defense, collagen synthesis, and immune support, and other biochemical reactions. So the answer is L-gulonolactone oxidase. Next question. Typhoid is caused by which of the following? The options are Salmonella typhi, Salmonella typhimurium, Salmonella paratyphi, Salmonella pullorum. Each of the options are Salmonella serovirus, belong to Salmonella genus. This question is one of the famous questions usually asked in the examination. Many of might know the answer. The answer is Salmonella typhi. Let's see the explanation. Salmonella genus is a member of Enterobacteriaceae family. Salmonella genus comprises of two main species. One is Salmonella enterica, the other is Salmonella bongorii. Do you all know the meaning of serovirus? Serovirus is the term used in the microbiology, which is short form for serological variant. This serovirus refers to distinct variation as subtype of your microorganisms. Each serovirus are distinguished based on differences in their surface antigen. If you take Salmonella enterica species, there are more than 2500 serotypes or serovirus. Both the terms are same. Okay. In contrast, the exact number of serotypes in Salmonella bongorii is less extensively documented, although it is known to exhibit lower diversity compared to Salmonella enterica. Among the Salmonella species, Salmonella enterica is the most pathogenic Salmonella genus that causes a wide spectrum of illness. Generally, the illness caused by Salmonella enterica is termed as salmonellosis. The severity of salmonellosis is dependent on the specific strain responsible for the infection and on the health status of the host. ranging from self-limiting illness that ceases in a week, that is, it may be acute enterocolitis to enteric fever, and severe invasive diseases which can result in hospitalization and even causes death. Deaths have been recorded, especially children below the age of 5. And even death also occurs in adults who are immunocompromised. In the Salmonella enterica species, there are many subspecies, the most importantly, subspecies 1 enterica, which accounts for 99% of the infections caused in human and animals. This subspecies 1 enterica's serovas are classified into 2 types based on the diseases they cause as well as the duration and severity of the diseases they associate with. One is typhoidal serovas, the other is non-typhoidal serovas. These typhoidal serovas are restricted to growth in humans only. Further, if you take typhoidal salmonella, it can be divided into 2 fevers, like one is typhoidal fever and the other is paratyphoid fever. So the typhoid fever is caused by Salmonella typhi. If you want to know what is a non-typhoidal serovas means, I have given enough details, just go through it. Next one, which of the following indicators is used in McConaughey agar? The options are phenol red, bromcrazole blue, neutral red, bromcrazole purple. In coming slides, we are going to see the usage of McConaughey agar, and we are going to see detail about what are the compositions and what are the indicators used. In the meantime, you try to figure out the answer. This McConaughey agar was the first solid differential media to be formulated, which was developed in the 20th century by Alfred McConaughey. This medium is a selective and differential media used for isolation and differentiation of non-fastidious gram-negative rods, particularly members of the family Enterobacteriaceae and the genus Pseudomonas. In microbiology, the terms fastidious and non-fastidious are used to describe different types of microorganisms based on their nutritional requirement for growth. Here is the comparison. Fastidious microorganisms means those microorganisms that have complex or specific nutritional requirement for growth. These microorganisms cannot grow on standard or general purpose laboratory media. In contrast to that, non-fastidious microorganisms are those that have relatively simple or broad nutritional requirements and can grow on standard or general purpose laboratory media. That means these microorganisms do not require any specific growth factors or any special condition for growth. From this, we understand that Enterobacteriaceae are non-fastidious gram-negative bacteria that can grow well under standard laboratory conditions. So, McConaughey agar is very simple and broad nutritional medium. Its composition are peptones and protease peptone, lactose monohydrate, bile salt, sodium chloride, agar, and distilled water. And one more thing is added to this is indicator. In McConaughey agar, 2 indicators are used. One is crystal violet, the other is neutral red. Crystal violet. This dye is indeed a selective agent that inhibits the growth of gram-positive bacteria, contributing to the selective properties of McConaughey agar. Neutral red. This pH indicator is a main differential component of McConaughey agar. It turns red at acidic pH level, that is below pH of 6.8, due to lactose fermentation and acid production by lactose fermenting bacteria. Non-fermenters do not change the colors of the indicator, remaining colorless. Together, these indicators contribute to the selective and differential properties of McConaughey agar. In the option, crystal violet is not provided, so the answer is only neutral red. Next question. In which of the following staining reaction acetone is used as a decolorizer? From the options available, we can come to a conclusion that this question is related to a staining technique. Other listed staining techniques are not familiar. So if in such a situation, directly go with the options which are you familiar, that is gram staining. Now see the explanation. In microbiology, staining techniques plays a crucial role in visualizing and identifying microorganisms under the microscope. Staining techniques serves several key purposes, mainly differentiation, identification, and visualization. Here are the most important staining techniques used in food microbiology. Among these, gram staining is the most popular and widely used technique. You know what? The staining techniques mentioned in the options like Ziegel-Nielsen staining, Lavetti staining, and so on are not specifically related to food microbiology. Now we are going to see detail about gram staining. The gram staining procedures involves 4 basic steps. The first one is primary stain using crystal violet. The bacteria are first stained with basic dye, that is crystal violet. Both gram-positive and gram-negative bacteria became directly stained and appears blue after this step. The next one. Following the application of the crystal violet, the slides is treated with grams iodine solution. This grams iodine solution serves as a mordant in this step, forming a complex with the crystal violet dye within the bacterial cells. This complex helps to fix the dye in place and enhances its retention by the bacterial cell wall. This allows the stain to be retained better by forming an insoluble crystal violet iodine complex. Both gram-positive and gram-negative remain purple after this step. Third step is decolorizer. In the context of gram staining, there are primarily 2 common types of decolorizers used. The one is acetone. The other is mixture of ethyl alcohol and acetone. Acetone decolorizer. Acetone is a strong solvent that effectively removes the crystal violet iodine complex from gram-negative bacteria. It is often used as a rapid decolorizer in the gram staining process, leading to quick differentiation between gram-positive and gram-negative bacteria. Acetone acts by disrupting the outer membrane of gram-negative bacteria, which is more permeable than the thick peptidoglycan layer of gram-positive bacteria. This permeability allows acetone to penetrate and extract the crystal violet iodine complex from the gram-negative bacteria. As a result, gram-negative bacteria loses the purple color of the crystal violet iodine complex and became colorless during the decolorization process. In contrast, gram-positive bacteria retain the purple color due to the tight binding of the crystal violet iodine complex within their peptidoglycan layer. So this is the decolorization mechanisms happening in the gram staining. Some laboratories may prefer the mixture, that is ethyl alcohol and acetone mixture. This mixture helps to slow down the decolorization process compared to acetone alone, providing a more controlled and gradual decolorization. This can be advantageous in achieving optimal staining results without over decolorization. The final step in the gram staining is counter staining with safranin. The mixture of ethyl alcohol and acetone is not given in the option. Only acetone is given in the option, so the answer is acetone. Next question. The mordant that is used in gram staining is, definitely you can answer this question. The answer is grams iodine. What is the meaning of the term mordant? While I am explaining about gram staining techniques, I forgot to explain about what is the meaning of mordant. Mordant is nothing but a substance that helps to fix or set a dye or stain onto a specimen. Okay. In gram staining, the mordant used is grams iodine. Final question. Types of botulinum neurotoxins produced by Clostridium botulinum. Clostridium botulinum is a very well-known organism. It produces 7 types of neurotoxins. If you know this statement, you can easily answer this question. Could you able to guess the answer? If you put numbers to the alphabet from A to G, there is 7 alphabets, though the answer is type A to G. Here we see the explanation. Biotoxins. What are biotoxins? Biotoxins are toxic substances produced by living organisms such as bacteria, fungi, algae, plants, and even animals. These toxins can have a various effect on other organisms, including humans, animals, and even other microbes. Biotoxins are often used by organisms as a defense mechanisms to protect themselves from predators, competitors, and environmental threats. These biotoxins are often referred to as naturally occurring toxic substances, shortly NOTS, because they are toxic compounds produced by the living organisms. There are several types in these biotoxins, and they are phytotoxins, that is plant toxins, marine toxins, bacterial toxins, fungal toxins, algal toxins. As Clostridium botulinum is a bacteria, we are going to see about bacterial toxins. Bacterial toxins are classified into two main categories. One is the exotoxins and endotoxins, based on their origin, structure, mechanism of action, and immunological properties. Let's see what is exotoxins. Exotoxins are proteins produced and secreted by bacteria, and they can cause harm to the host cell and the tissues. Both gram-positive and gram-negative bacteria have the capability to produce exotoxins. Further, the exotoxins can be classified into many types. Some are enterotoxins, neurotoxins, cytotoxins. See, the neurotoxin, Clostridium botulinum produces neurotoxins. Okay? Now you all can easily understand. That is, Clostridium botulinum is a gram-positive bacterium. It produces 7 distinct stereotypes of botulinum neurotoxins labeled as BOTN A, B, C, D, E, F, and G. Each serotype has different properties and can cause botulism under specific circumstances. However, not all serotypes are equally common or equally implicated in case of botulism. Among the 7 serotypes, A, B, E, and F are the most commonly associated with the human botulism. Botulinum neurotoxin C is less frequently reported in human botulism, while botulism neurotoxin D and G are rarely causing botulism in humans. That's enough with the exotoxins. The other bacterial toxin is endotoxin. These endotoxins are part of the cell wall of gram-negative bacteria and are released when the bacterial cells are lysed or destroyed. These endotoxins are composed of lipopolysaccharides. That's all for today's video. In Food Catalyst, our team is extensively working on the explanation part. Don't skip even a single word. At this point, I am signing off, and this is Dhilip. See you on the next video.